Hi everyone, welcome back to the Coral Conservation Series with the Texas State Aquarium. I'm Dr. Keisha Bahr, and we are back from Hawaii. And with coming back from Hawaii, we've brought quite a bit of samples to be processed, and we're continuously processing the samples and processing the data and learning more about how these corals are growing under different chemistries of ocean acidification. Also, at the same time, we're really trying to understand how corals are growing in general. One of the really cool things that we have seen so far is one of our species of corals continues to grow even though we do place it and grow it in under ocean acidification scenarios, and it continues to grow. But one of the things that we're really unsure about is if it's still growing and having a thick skeleton at the same time, or are they becoming more fragile? So some of the work that we're doing is really trying to understand is how dense is that coral skeleton underneath these different coral or ocean acidification chemistries. All right, so we brought back the coral samples from Hawaii. We have thousands of samples of small fragments of the corals that we've been working with. And with that, we removed the tissue as we showed in our last video. And with the tissue, what we're trying to really understand is how many symbionts or zooxanthellae or algae that live inside the coral, how many do we see in each of those different fragments. Also, we want to see how much chlorophyll those algae have, because chlorophyll is really important to understand because that's how the algae use sunlight to gain the energy that they then give to the corals to help sustain the corals over time and over their growth that they have. So what we're doing here is we have removed the tissue from the coral. We then have this placed in a smaller tube, and we extract the chlorophyll using acetone, which is safe for us to handle and use, but it also allows us to extract that pigment so that we can measure that pigment in a spectrophotometer that you will see here. And we measured those wavelengths at 2 wavelengths where we know is the maximum amount of absorbance that would happen with chlorophyll. And we quantify those measurements doing calculations after we get these 2 different wavelength measurements. We also do counts of the symbionts that we have, and that allows us to see of how many zooxanthellae or how much algae the corals have. And we do a ratio to look at how much chlorophyll is there per coral and how does that relate to the amount of zooxanthellae or symbionts they have. And then the other thing that you see us doing here is actually looking at the skeleton. So I mentioned previously that we stain the coral so that we want to see where the corals are currently at, and then afterwards we want to see how much they've grown after they've been in these conditions for 30 days. With the stain, it's this really beautiful pink color, and anything after that pink color or that white skeleton that you see, that's the amount of growth that we've seen of the corals. And this is what we consider to be called linear growth, or corals growing upwards, kind of like how we grow and your mom or your dad might mark it on the wall, and that will tell us like how much they're growing in a certain amount of time. But then also we want to look at the skeleton and see how dense that skeleton is. And this is a really simple process. It's something that you probably learned during your science classes, is how do we measure density. What we do is we place the corals in water, we make sure that they're very full in saturated water, and then we place them inside of a tube and measure how much displacement or how much the volume of water changes based on how big the coral is. And from that we can do the mass of the coral divided by the volume to get the density of the skeleton of the coral. And this kind of gets back to this really cool thing that we're hypothesizing, or we're making guesses to see how corals are growing differently under these ocean acidification conditions. Is we think one of the corals might continue to grow upward or linearly, so have that sustained white growth. And with that, we think that they might be compromising how dense their skeletons are. So even though they're continuously growing, we think that their skeletons are becoming less dense, meaning that they're more fragile. So this is still an issue, because if we want our corals to continuously grow these structures and also provide a barrier for us here on land, if they're more fragile, then they're going to break down and they're going to crumble a lot easier in our future conditions. So we're continuously looking at that. We're doing tons of these measurements, and we're hoping to have the results of that very soon.
So my name is David Armstrong. I'm a current PhD student researcher right now in the Bahr Lab. Before I did grad school, I used to build aquatic life support systems for large industrial aquariums and zoos, engineering and construction work, and it was really great. I loved it, but I wanted something a little bit more, and so I found Dr. Bahr and her lab, and I reached out to her, and I was able to come visit everybody, and I loved the community and the area, and it just all felt so welcoming. So that's kind of what drew me here and drew me to doing this research, and especially with the projects that we were working on were really interesting, really applicable, and yeah, I was just really excited to do it.
Yeah, so I'm Brianna Edetta. I'm a first-year master's student within A&M Corpus. I chose mechanical engineering for my undergrad because growing up I was really into math. I really love math, especially just solving a problem, and when you finally get it after trying it so many times, it's just a really great feeling. My dad is also a mechanical engineer, so he kind of also inspired me to go towards engineering and seeing how he applies it just in everyday life, not even just in his job. It's something that I really enjoyed and something that I just wanted to go for. When I was thinking about what I'd want to do after graduation, so many of my friends were just going to go into industry, they were going to start their full-time jobs or continue with an internship that they had started, and that wasn't something that I really wanted to do because it didn't spark any joy in me, and that's something that I really want. Is whatever work I do, I want to be happy that I'm doing it, and I want it to be able to be something that's beneficial for other people as well, not just something that's going to bring me the most money or whatever any other reason may be. And when I really started thinking about it, something that I've always really enjoyed studying, just on my own, is marine biology. I really love those sections within my science classes when I was in high school, or even when I took a biology class in undergrad. I really enjoyed just learning about the different functions that are needed to even just support life. And I found Dr. Bahr, found her lab, and I just thought that what she was doing was something that was really great and amazing, and I just really wanted to be a part of it. And funny enough, I actually didn't know much about corals before starting my master's degree, but now it's I'm able to write my literature review, and looking back on it, it's like I wouldn't have been able to do this a year ago. So it's crazy to see how much you learn even just by reading or by listening.
Hi, my name is Ignacio Reda. I'm an undergraduate researcher here in the Bahr Marine Ecology Lab. And last year I went to Hawaii. I was super excited. I thought I knew what I was getting myself into, but I did not know what I was getting myself into. It was so much more intense than I could have ever have imagined, but so rewarding. I felt like it was just such a nurturing environment, learning from all these amazing scientists and also stepping into my own way of doing science as well. I thought it was very rewarding, and actually having the chance to just do experiments, learn more about global stressors such as ocean acidification, which is such an amazing experience.
My name is Megan Humphrey. I am a post-baccalaureate research intern in the Bahr Marine Ecology Lab. Yeah, no, Hawaii was great. It was my first flight, so being an 8-hour long flight was kind of intimidating for the first time, but the flight was great. Hawaii was an amazing experience. Just being hands-on with the coral is just knowing how they start out was really awesome. Because when they come here, they're already in tiny fragments. We're just processing them, airbrushing them, doing all that stuff with them. But seeing the coral colony before it's fragmented and seeing the experimental treatments on it, that was amazing, because it kind of came full circle with what we were doing, and it all kind of clicked together, and the data made more sense. Seeing in person. And the people in Hawaii were great. Everyone was just so friendly, and the scenery was beautiful. I definitely would love to go back. It was amazing.
My name is Margaret Harp. I graduated from TMUCC in 2021 with a bachelor's in environmental science and a concentration in marine and coastal resources. So I wasn't exactly on the marine biology track, kind of studied a more broader approach to coastal and marine environments than just specifically marine biology. Hawaii was pretty amazing. Getting hands-on experience solely focused on just the NSF experiment. It was awesome. We got, hands-on with corals. We got to help collect them. We helped run the experiment, like 26-hour experiments, like a whole 14-hour shift, at night and stuff. And that was really good experience to have, because it's, there's so many things that can go wrong, because it's not just a controlled environment. We're outside, and the rain, sun, we were working through all of that. So that was definitely helpful to see how rugged it can be, and the people there were amazing, really helpful. Like, I hadn't had experience with a lot of the equipment that they had there, so I had to learn a lot on the spot and work with that really quickly. And it was really amazing to see that after we've done a lot of work here in Texas, we actually got to go see what the corals looked like before we got them.
Future career aspirations. Well, I am looking into going to graduate school. I've actually been accepted to the University of Puerto Rico, so hopefully I'll be starting either in the fall. What drew me into Puerto Rico had a lot to do with corals themselves, and so I know this is gonna be long-winded, but I love corals specifically because it's an individual polyp working together in like a colony, and I guess I kind of see myself as a polyp working in a community, and so Puerto Rico, I went there last year for a conference. Love the environment, love the work they're doing there, and just seeing myself in the people themselves is something that I really responded to. And I searched for a PI. His name is Dr. Travis Courtney. I spoke with him, loved his energy. He's a lot just like Dr. Bahr. I don't know, just nurturing and very loving and very intelligent. So I really love that about him, and I just saw a lot of Dr. Bahr in him, and I saw myself really growing under his tutelage. So that's really my main reason for going to Puerto Rico, is just to work with him and work with the corals there, and hopefully learn to bridge the gap between the people and the coral reefs there. So I'm very excited about it. It's the next step, and all that was possible through, of course, my hard work and all of the just the camaraderie or nourishment I've got from being in this lab. So I'm very thankful to Dr. Bahr and all my colleagues here in the Bahr Marine Ecology Lab.
I've actually reached out to several graduate schools. I think I reached out to like 20 professors. 3 of them got back to me and said yes. And I've actually decided to go to Louisiana State University and work with Dr. LePere there in the School of Renewable Natural Resources. And I really feel like I wouldn't have had that opportunity if I didn't get the experiences that I did here as a post-bac. And also I wouldn't have known about it unless I was in this lab, because it was in like our lab chat channel that someone posted that Dr. LePere was looking for, research students, and I just happened to email her and got the position. So yeah, I feel like this position's been very beneficial for, myself personally, my growth as a scientist, and just like overall it's been a great experience.
I think some advice that I would want to give to either people who are in high school or even early within their college careers, to not be afraid to do something that might be different than everybody else. Transitioning from engineering to marine biology, all of my friends and even some professors were telling me to not do it, to wait a bit, to go into industry, have them pay for my school and everything, and make sure that this is something that I'll actually be able to apply engineering towards. But I knew that if I waited any longer, then I would start being comfortable where I am. And you always hear like comfort is the enemy of like progress, or comfort is the enemy of growth. And I really wanted to see what all I can do. I didn't want to just remain within engineering. I really wanted to broaden my field. So I would say to, definitely just not be afraid to kind of go against what other people are thinking or what other people are saying, and also just to be curious. Like, don't be afraid to ask questions about different things, or if there's something that you're really interested in, but your degree is, or your study is, within a very small specific field. I would say to try and find how you can apply your knowledge or your background towards whatever it is that your passion is. So
if you are having a hard time deciding, you should definitely network, talk to your peers, talk to your professors. There's so many podcasts out there that can help you learn like what it's like to be a marine biologist, what it's like to do conservation. If you like to get into diving, there's so many helpful resources out there for that. But if you love the ocean, you're already kind of in it, so it's something that you'll continue to love.
When I was starting to learn more about research and understand science, and actually my journey as an undergraduate degree, it was really challenging for me because I felt like I didn't know a lot of things and I didn't really know who to talk to. A lot of that comes because I was a first-generation college student, so I didn't really know how to navigate academia or navigate college, or really know how to get involved in things, and I was really nervous about asking about how do I get involved in research, or how do I get these experiences that are required of me to go into the field that I wanted to do. So having those anxieties and that unknown of really understanding what do I do or who do I talk to, I think really rooted my mentorship style, because I understood what it was like to be in a position of not knowing these things, or feeling like something was wrong because I didn't know how to approach things. So with my students, I have had a very hands-on approach in helping them navigate the uncertainties that they might be facing, especially here at Texas A&M Corpus Christi, where a large majority of our students are first-generation college students. Alongside that, I was a recipient of valuable mentorship, and I understand the importance of what can happen if you do have a mentor who does truly care about you and who truly wants to see you be successful. So just being exposed to that and having that in my daily life has really shown me that you can go really far if you have somebody who, one, believes in you, and two, who can help you navigate these things that you might not know. And that is something that I strive to do every day with my students, because there is so much potential, and I'm just trying to help them unlock the potential that I see in them, so that they can see it in themselves, so that they can achieve their goals. All right, so this summer we're heading back out to Hawaii for the final set of experiments that we will be doing for this project. Also, at the same time, we do have some students here in Texas who will be continuously processing these samples, because it takes quite a bit of time to go through all these samples. But while we're in Hawaii, this summer we're going to take 6 new students to the Hawaii Institute of Marine Biology. And what's really fun and exciting about this is we also have some of these students who are biology focused and environmental science focused, but we also have students who are engineering focused as well. So I'm really excited about this summer, because all 6 of us will be working together, answering and constructing some really cool devices to help us understand how corals are growing. We will also still collect those coral samples and bring them back to us in Texas. So we will be in Hawaii this whole summer, and when we get back, we're excited to touch base and let you know how the summer went, what kind of samples we got, and what kind of things did we construct to help us answer some of these questions that we have.